So yes, I thought I would share some experiences that we've had with organizing these more advanced single cell courses. And this is experiences that we've done like partly alone at the Elixir Switzerland and partly together with the Elixir Sweden as well. So the two courses that this is based on have been given the last 2 years. So the first one was in May 2020. So this was a 3-day course. It was online streamed, but otherwise it was a fairly, let's say, standard course in containing lectures and exercises on different topics. And we were covering partly some technical topics, so not directly related to specific single cell applications, including how to work with Python and R together, and also how to work with on-disk data, so HDF5 files, for example. And then we had a couple of single cell specific topics. So we talked about quantification of gene expression, so other ways than just running Cell Ranger. We talked about RNA velocity, spatial transcriptomics, and deep learning. The other course that you've already heard a little bit about from Paulo was a project-based course. So this was from this year, August, September, and this was, well, a different setup, right? So there was a little bit of technical basic information in the beginning, so to make sure everyone was more or less on the same level. And then the majority of the time was really spent on group projects. And the topics we had there was, as you already saw in Paulo's talk, the spatial transcriptomics, RNA velocity, multi-omics integration, and deep learning. So to kind of talk a little bit about how we selected the people here, we had a lot of demand for attending these courses. We could only accept less than half of the people who applied for each of the courses. We did select people based on a short survey. So we asked about their previous experiences with single-cell RNA-seq and also with Python and R, for example, about what kind of projects they were working on, their motivation to follow the course, what they thought they wanted to get out of it, and so on. And then the selection was based on that. For the project course, we also allowed people to select or to give a preference for which of the projects they would like to follow. And I think this meant that we got a fairly homogeneous population of participants in terms of experience, right? So they still worked on, of course, very different projects from different countries in different environments and so on. But in terms of experience, it was reasonably homogeneous, which of course made the whole developing the course much easier than if the population is very heterogeneous. We also, for the projects course, we did allow everyone who applied to attend the introductory talks of the different topics and also the project presentation in the end of the course. And both courses the lectures were recorded and put on YouTube for everyone afterwards. In terms of the topics, as I said, it was quite different between the two courses in terms of how they were set up. The 2020 course was basically half a day per topic. And as I said, we had the technical topics, the Python and R and the HDF5, working with HDF5 files. And then we have the specific focus topics on single-cell RNA-seq and single-cell omics. And I think there is a trade-off here between trying to cover the hottest topics and at least having a somewhat best practice workflow to teach. And I think my experience is that for these kind of courses where you don't have that much time per topic, it helps to have at least a reasonable best practice workflow that you can teach. For the project course, it was a bit different. So here the students had much more time for to spend on each of the topics. So we could work with much more open-ended questions, much more room for exploration, trying different tools, different ways of solving a problem, different ways of maybe even asking the question. So in that sense, I think it was a bit of a difference in what type of topics we could actually cover, or which types of, how you would approach a specific topic, let's say. So all groups had to present their project at the end of the course as well, so everyone could at least get something out of the topics also that you didn't work, the top project topics that you didn't actually work on. On the computational setup side, we really did not want to spend a lot of course time setting up and troubleshooting computational environments, because it was an advanced course, we assumed that people should have already had some experience working with single-cell RNA-seq. So probably they already, or hopefully they already have a working environment at home. So we didn't want to spend a lot of time on that. So in both cases, we actually run the analysis or the exercises on remote platforms. The 2020 course was run on Renku, which is a platform from the Swiss Data Science Center. And basically what that required from us was to create a project. You can get the link to the project here, it's open. Basically, we need to create a Docker image where that's based on a base image from Renku that has all the infrastructure. We installed the R packages, the Python packages, and then what the students had to do was to get an account on Renku, fork the project, and then basically they had all the material and could start instances where they could run analysis as well on the Renku platform. So you would have access to RStudio and the Jupyter Notebook and the terminal based on this same underlying container. For the 2021 project course, we instead used AWS cloud instances. So these were all set up by Gert, who is here. So all the technical questions he can answer if you have any. So this was kind of similar, right? It's based on Docker containers as well. We pre-installed all the R and Python packages that we needed. And then you would as well have access to RStudio and Jupyter Notebooks, but in this case, it was actually via different containers. But if on the AWS instance, you could actually access the same working directory and data directory from each of these interfaces. And the nice thing here was that we could also, for the deep learning project, we could set up instances with GPU access. I think generally this works well. It freed up lots of time that we would otherwise probably have spent troubleshooting installations. Maybe not very surprising. The more you can test things in advance, the better. Especially things like memory requirements, making sure that your instance hopefully doesn't crash if people go a little bit like away from exactly what you have in your exercise document. If people try out different things, run multiple different pipelines to try to still stay below the memory limits of the instances. Also trying to make the image as complete as possible from the beginning. We did install a couple of things during the course, which works, but it makes things a little bit more complicated, and some things have been updated and may not be compatible with what you installed before and so on. So, the more you can prepare, the more you can test in advance, of course, the better. And I think it was really helpful in both of these courses. We actually had someone with really detailed technical knowledge there during the course. So Gert was there for the second course, and we had people from the Renku team available on Slack and also giving a short talk in the first course. So the cost for this I got from Gert as well. So for the AWS instances for a week, the CPU instances were about $10 per participant for the whole week. The GPU instances were quite a bit more expensive, so that's up towards $60 per participant for the whole week. So to kind of summarize some other challenges and things we have learned, I think that especially for these advanced courses, even if the students have the same level of experience, they will for sure be working with different frameworks. Some people will be used to ScamPy, some people will be working with Seurat, some people will use Bioconductor. So maybe to a larger extent than in an introductory course, you kind of need to provide avenues to do the analysis from each of these starting points, let's say. And if that is a whole workflow in each of these frameworks, or if it's really just taking some time to tell people how you can convert from one to the other, right? But so that people can get it into the world that they are familiar with. The technical topics that we included, so the Python and R, the HDF5 data technical issues, they were appreciated by the participants because maybe they're not included very often, but you tend to run into these issues when you have to actually analyze your data yourself. What we did in both of the courses was to actually reach out to developers of packages that we wanted to cover and engage them as teachers. So we had PhD students and postdocs who were developing some of the packages that were covered in the courses. to teach, and this works really nicely. I think you get I think it's a great way to cover really emerging topics by people who are really immersed into this and know all the practical details and all the issues and all the challenges. As I said, providing the pre-configured cloud instances really helped us to free up time to focus on the actual course topics. And I think we noticed when comparing the courses that this project-based setup did lead to a higher engagement from the students, both with the topics and with each other. But of course, you get direct exposure to fewer topics, right? You get something out of all the topics, but you get a deeper exposure to a smaller set of topics. So that was it. Thank you for listening, and these were all the people I hope who contributed to these courses.